Modern pharmaceutical development is an exercise in failure. For every compound reaching the pharmacy shelf, laboratories discard thousands of failed chemical assay plates. The traditional process of discovering a new drug from scratch takes between 10 and 17 years. The financial toll for a single novel drug stands at $1.8 to $2.6 billion. Despite that massive investment of time and capital, the success rate for approving a new molecular entity stands at a dismal 2.01%. This high attrition rate forces the pharmaceutical industry to seek highly efficient alternative methodologies, rather than continuously trying to reinvent the wheel with every new disease target. The traditional development model is mathematically unsustainable. There is now an urgent mandate to extract new therapeutic value from existing pharmacological assets. This 2021 review article by researchers Sanae and Sheikh codifies how the industry is transitioning drug repurposing from a series of accidental discoveries into a formalized, highly structured pipeline. Drug repurposing, also known as reprofiling or therapeutic switching, is the deployment of already approved, shelved, or discontinued drugs for novel alternative disease indications. This flowchart shows the traditional drug discovery process. You can see the lengthy, distinct phases stretching out across that 10 to 17-year timeline. Repurposing completely bypasses those early, wasteful phases. Because these are existing molecules, researchers already have access to robust clinical data. They can jump straight from compound identification into late-stage clinical trials. The measurable impact of this bypass is striking. The timeline shrinks to 3 to 8 years, and the programmatic completion rate jumps to over 65%. Repurposing operates as a primary, high-yield strategy. It uses historical safety data to systematically eliminate early-phase risk, capitalizing on years of prior research. Modern repurposing has evolved far beyond waiting for accidental discoveries. It is now a deliberate, hypothesis-driven science. The core biological mechanism enabling this efficiency is polypharmacology. This is the ability of a single drug molecule to interact with multiple distinct targets or disease pathways in the body. The average drug is known to interact with 6 to 13 different biological targets. In this context, what we normally consider off-target side effects are actually untapped therapeutic opportunities. This diagram outlines the workflow used to identify those opportunities, starting with an existing library of approved compounds. Instead of physical assays, scientists save time by running a virtual screen, using computational models to predict how an existing drug engages a new target. Polypharmacology provides the necessary underlying biological potential, but accurately identifying up to 13 hidden targets across thousands of existing drugs requires a massive computational filter. The primary challenge of computational screening is that biological data is highly fragmented across the field of genomics, the study of transcriptomics, the map of metabolomics, and deep proteomics. To solve this, researchers integrate disparate chemical, biological, and genomic databases into a unified architecture. Semantic network inference maps structural relationships between entities, like how drugs link to diseases. Machine learning algorithms then analyze this network to predict novel links too complex for humans. Semantic network inference acts as a computational compass, transforming billions of isolated data points into targeted, highly probable clinical hypotheses. Identifying a new target is only half the battle. Despite these scientific advances, bringing a repurposed drug to a patient requires navigating the severe economics of late-stage development. Researchers frequently encounter the financial valley of death. This is the monetary gap between successfully modeling a repurposed drug and securing the massive funding required for Phase 2 and 3 clinical trials necessary for FDA approval. Traditional pharmaceutical companies generally lack the financial incentive to fund these trials. If a drug is already off patent or was previously shelved, a company cannot secure exclusive rights to recoup their investment. Without an external catalyst, the most brilliant computational discoveries will permanently stall in the laboratory before ever reaching a patient. Recognizing the massive public health potential, governmental agencies have stepped in to bridge this financial gap. In 2012, the National Institutes of Health created NCATS, the National Center for Advancing Translational Sciences. Its specific mandate is to fund repurposing efforts from early in silico projections all the way through late-stage clinical trials. They provide highly specific support mechanisms, such as the TR&D initiative, which actively helps researchers assemble the complex regulatory data packages required to get shelved drugs approved for trial entry. This is an international strategy, supported by equivalent organizations globally, such as the Canadian Institutes of Health Research. Integrating big data networks, targeted combination therapies, and institutional funding has transformed repurposing into a rigorous engineering discipline. This system provides a scalable solution to the pharmaceutical industry's most unsustainable inefficiencies.